My name is Manuela Yassa, and I serve as Director of Outreach and Education at the Center for the Neurobiology of Learning and Memory here at UC Irvine. This week, you may have heard, is Brain Awareness Week, a global celebration led by the Dana Foundation to increase public understanding of the brain and the role that neuroscience plays in our everyday lives. It's a time when neuroscientists around the world create opportunities for meaningful dialogue between science and society. Here at the Center for the Neurobiology of Learning and Memory, you all know that we often say that every week of the year is Brain Awareness Week because building those bridges between science and the public is central to who we are. We believe that it's not enough to do the science. We also have to share the science with the world. And just as importantly, we have to listen to you. Science is stronger when it's connected to the communities that it serves. The questions that shape research don't just come from within the lab, right? They emerge from lived experience, from the curiosity of the public, and from the real world challenges that people face every day. And that's what tonight is all about. This series, How It's Made Neuroscience, is part of that broader effort. It's our way of opening up the process of science, making it more visible and more human and more accessible. And tonight is not just about the scientists and what they discover, but how they get there, right? The questions that they ask, the challenges that they navigate, and the uncertainty that they encounter, and of course, the curiosity that keeps them going. Of course, this space is for you to ask questions, to be curious, and to be part of this conversation. Before we begin, I want to recognize the incredible team that is making tonight possible, starting with my team at the Center for the Neurobiology of Learning and Memory. Dr. Erin Conway is here and Dr. Morgan Coburn. And the event tonight was thoughtfully organized by our graduate student leaders, and we're joined also by a group of ambassadors who will be helping to moderate the questions as well. The leaders that have organized tonight's event are Kate Sormis, Kristen Vallejo, and Tracy Uzoigwe. They're not only outstanding scientists, but they are also deeply committed to connecting science to the broader community. I'd also like to thank our panel of faculty who are here with us participating, and of course, you're going to get to know them. And finally, thank you to you, to our community. You always show up with so much curiosity and ready to engage with us, and I'm really excited for a wonderful evening. And with that, I'm going to turn it over to Kate to get us started.
Thank you so much, Manuela. Thank you for introducing us. I want to thank you all for being here and participating today. We did a similar event last summer, so if you're joining us again, welcome. Thank you so much for being here. And if this is your first panel, I hope you enjoy it. My name is Kate Inman-Sermis, and I'm a PhD candidate here at UC Irvine, and I've spent the last several years studying the brain's immune system, but I am here with you today as one of the co-chairs of the CNLM Adult Outreach Committee and the moderator of today's virtual panel event. I've been interested in bridging the gaps between the scientific community and members of the public pretty much as long as I've been interested in science. And right now, I think transparency in science feels more important than ever. So for that reason, we thought that talking to you all about what neuroscientists actually do all day and letting you ask them some questions would be a great place to start. So before we begin, I have a few housekeeping details to go over. The questions that we will be asking our panelists today were generated based on the questions that were submitted to us during your registration. And we hope to cover as many of them as we can while discussing the process of actually doing scientific research. But we do want to note that very specific questions about particular diseases or neural mechanisms might be harder for us to answer in this format. It's more of a how it's made conversation. And as we answer these questions, feel free to ask additional questions using the Q&A feature in the Zoom in the Zoom window. And our moderator team will help us keep track of those questions, and we will do our best to circle back to them as relevant. And we have a more open Q&A session also planned for later in the discussion. We also wanted to say that this is a panel geared towards adults in the community who want to learn more about research and the scientific process. We did get some questions from high school students wanting to get involved with research or to learn more about the brain. And so we want to invite you to visit the CNLM website to explore the research being conducted at the center or reach out to the CNLM Office of Outreach and Education for some of our programs that are involving the youth. And our mods will be copying and pasting the email that you can reach out to in the chat here. But everyone will have a chance to hear about our panelists' journeys into their research careers, which we hope will be insightful for any current students who are part of our audience right now. We want to remind you of the CNLM Code of Conduct, ask that everyone remains respectful. And finally, we are not a clinic and cannot provide diagnoses or healthcare advice. We are a resource to bridge our community with neuroscience research taking place at UC Irvine. With that, I would like to introduce you to each of our panelists and bring you all behind the scenes of their work with some short videos to show you our researchers and their lab spaces. So first, we will start by introducing you to Dr. Sara Mednick, a leading cognitive neuroscientist at the University of California, Irvine. Dr. Mednick's work has fundamentally advanced our understanding of how sleep supports memory, learning, and cognitive function. Her research integrates behavioral, physiological, and computational approaches to uncover the mechanisms by which sleep, particularly features like sleep spindles, facilitates memory consolidation and brain health across the lifespan. She has received numerous prestigious awards, including funding from the NIH, NSF, DARPA, and the Department of Defense, and her work has been widely recognized both within the scientific community and in major public media outlets. Dr. Mednick also leads the Sleep and Cognition Lab at UCI, where she mentors the next generation of neuroscientists and continues to push the boundaries of sleep science. So please welcome Dr. Mednick, and let's go behind the scenes into her work with a video.
What happens in the brain while we sleep? As we rest, the brain moves through different stages that help turn new experiences into lasting memories. In the Mednick lab at UC Irvine, researchers are trying to understand exactly how sleep strengthens memory and what kind of sleep, short power naps or longer naps, that include all stages of sleep are most effective for enhancing learning. To do this, they study how different stages of sleep affect learning. First, participants complete a virtual reality navigation task in the morning of the experiment. Here, participants explore a simulated environment and learn to navigate through it, similar to learning your way around a new city. Along the way, visual cues help guide them, and allowing researchers to assess how well the participant is learning spatial information. Then, in the early afternoon, researchers carefully prepare the participant for sleep monitoring. An electroencephalogram, or EEG cap, is placed on the head to record electrical activity from the brain, allowing scientists to identify exactly when someone enters different stages of sleep. To ensure a clean signal, researchers first clean the scalp before applying small amounts of conductive gel to each electrode. Once everything is calibrated, the participant rests in a quiet nap room, where researchers track their brain activity in real time. Participants are assigned to one of three groups, where they may take a 60-minute nap, a longer 90-minute nap, or no nap and remain awake. Comparing these nap lengths allows researchers to examine the role of REM sleep, the dream-associated stage that tends to occur later in sleep, in spatial learning. Early results suggest that different stages of sleep contribute in unique ways to memory consolidation. Both REM sleep and non-REM sleep appear to play important roles in strengthening the brain's internal maps of space. By combining behavioral experiments, virtual reality, and brain recordings, the Mednick Lab is uncovering how sleep transforms new experiences into lasting memories. These discoveries are helping scientists understand why sleep is one of the brain's most powerful tools for learning.
Great. Thank you so much, by the way, to our amazing CNLM marketing team who has put together these videos. We did this last year as well, and they just have done such a good job. So I'm excited to see the other two. I would like to then move on to Dr. Lisa Flanagan, who is an assistant professor in the Department of Neurology at UC Irvine. She holds a bachelor's in zoology and psychology from Duke University and a Ph.D. from the neuroscience program at UC San Diego, where she conducted research on Alzheimer's disease. Following postdoctoral training at Brigham and Women's Hospital at Harvard Medical School, she joined UCI, where she now leads a research lab at the intersection of neural cell biology and bioengineering. Dr. Flanagan's research focuses on understanding how neural stem cells make fate decisions to determining what types of cells they turn into, with the ultimate goal of improving stem cell transplantation therapies for conditions such as spinal cord injury, Alzheimer's disease, and stroke. So without further ado, let's move on to Dr. Flanagan's video.
Every cell in the brain has a job. Neurons send the electrical signals that shape our thoughts. Glia support a healthy brain environment and protect those neurons from injury. But where do all of these cells come from? Inside the developing brain, neural stem cells act like biological factories. They divide and transform into the many different cell types that make up the nervous system. In the Flanagan lab at UC Irvine, researchers are studying how these stem cells decide what to become, and how those decisions might help us repair the brain. First, the scientists grow neural stem cells in carefully controlled culture conditions. Over time, the cells multiply and expand, creating enough material for experiments. Researchers then examine the cells to see which proteins they produce, tiny molecular markers that reveal whether a cell is becoming a neuron, an astrocyte, or something else. Understanding these differences is especially important for diseases like glioblastoma, an aggressive brain tumor that often resists chemotherapy. To study these subtle molecular differences, the team combines biology and engineering. They build tiny devices called microfluidic chips. These chips are made from a soft polymer called PDMS that is molded and bonded to a glass surface to create microscopic channels. Once assembled, the device is attached to an electronic chip and examined under a microscope. Now, the experiment begins. Cells are introduced into the device and flow through the microchannels one by one. Because cells carry electrical charge, they move in response to small electric fields inside the chip. Cells with different surface proteins behave slightly differently, changing how strongly they move through the channels. By observing where the cells travel, researchers can separate, reculture, and expand the numbers and analyze the different populations of neural stem cells and their descendants, or drug-resistant glioblastoma. These measurements help scientists understand how brain cells develop, how tumors like glioblastoma evolve, and how the brain might someday be repaired. By combining cell biology, engineering, and advanced imaging, the Flanagan Lab is uncovering the hidden rules that guide how brain cells are made. These discoveries could one day help scientists rebuild damaged brain tissue, one cell at a time.
All right, great. And also a quick clarification. I was using an out-of-date bio to write my bio for Dr. Flanagan. She is a full professor here at UCI. There is no way an assistant professor could be as prolific as she has been in her career. All right, with that, I would like to move on to Dr. Jeffrey Krichmar. Our next panelist is a professor in the departments of cognitive science and computer science here at UC Irvine. He earned his Ph.D. in computational sciences and informatics from George Mason University and completed his postdoctoral training in theoretical neurobiology at the Neurosciences Institute. At the Cognitive Anteater Robotics Laboratory, or CARL, his team designs robotic systems guided by large-scale simulations of the mammalian brain. By embedding these models in real-world environments, his work provides a powerful framework for understanding how neural systems give rise to behavior. His work spans everything from spatial and episodic memory to motor control and perception, using neurorobotics to bridge brain, behavior, and environment and offer new insights into brain function. So let's welcome Dr. Krichmar and proceed with his lab video.
How do animals move through the world so effortlessly? They sense their surroundings, make decisions, and navigate complex environments with ease. In the Cognitive Antita Robotics Lab at UC Irvine, researchers draw inspiration from nature to design robots that can sense, think, and behave like animals. These robots can help us understand how biological brains process information and can guide the design of artificial intelligence systems. Rather than studying intelligence in isolation, the lab builds robots that interact directly with the world. In these systems, intelligence emerges through the interaction between a brain-inspired controller, a physical body, and the surrounding environment. One example is the Octobot, a robot inspired by octopuses. Octobot explores its environment using touch. Sensors allow it to physically feel objects around it and respond to what it detects. Researchers design the robot's hardware and software, from CAD models to neural-inspired algorithms that allow it to interpret sensory information and decide how to respond. By combining tactile sensing, behavior, and visual displays, researchers can study how perception and communication emerge from the unique sensory systems of octopus. Another robot is the Cuttlebot, a robot inspired by cuttlefish. The Cuttlebot navigates its environment using LiDAR sensors, which allows it to detect obstacles and map the space around it. Beyond navigation, researchers can program the robot to display a delayed gratification behavior when choosing between different rewards. When faced with an immediate but lower value food reward, the robot can learn to wait longer to receive a higher value one. This ability to delay an action in pursuit of a better outcome is an important part of intelligent decision-making and a behavior seen in children as young as 5 years old. It also has been observed in the robot's namesake, the cuttlefish. Studying how robots learn to balance these choices helps scientists understand the biological roots of decision-making and design machines that can adapt their behavior based on changing rewards. The lab's Carl Jack robot takes these ideas into the real world. This mobile robot can navigate outdoor environments while weighing multiple factors at once. Sometimes the best path is the fastest route. Other times, it's the path that uses the least energy, especially when its batteries are low. To solve this problem, the robot learns from experience. With each journey, its internal networks adjust and strengthen, a process similar to plasticity in the brain. Over time, the robot develops navigation strategies that resemble those used by animals. Its internal maps can even produce activity patterns similar to place cells, the neurons in mammalian brains that help represent location in space. By combining these internal maps with real-time sensory cues, the robot can understand where it is in the environment, often more reliably than GPS alone. Inside the Cognitive Antita Robotics Lab, robots aren't just machines. They're scientific tools that help researchers explore one of the most fascinating questions in neuroscience. How do we perceive the world, make decisions, and learn from experience? By building robots inspired by biology, scientists are uncovering new principles of intelligence and bringing us closer to machines that are truly cognitive.
Robot waving goodbye. That was a really nice closer there. Thank you all for being here. Thank you for participating in these videos. I hope they were a nice introduction for everyone into each of your worlds. And I love the broad array of work that we have represented here with each of your research programs. It's so cool to have such diversity. So without further ado, let's go ahead and get into the question section. And I also want to remind you all to, if you have questions that have come up after watching those videos, or if you get inspired to ask anything as we're having a discussion, you can use the Q&A box. We don't have the ability to call on anyone who has a raised hand, but we can answer questions that come up in the Q&A. So if you have anything come up, send it our way. So I want to ask everyone to start. One of the great things about being a neuroscientist is every day can look a little bit different. So, what does an experiment day in your lab or in your research subfield look like? And do your experiments require a ton of scientists? What are the logistics here? And I think we can start with Dr. Flanagan.
Hi. Well, thank you so much. And I want to just start by thanking everyone at the CNLM for all the work they put into this series. It was really fun to see the videos from the other labs. So thank you so much for putting all that together. And I'm really happy to be here as part of this panel. Yeah. So, as you saw kind of from the video, my lab works a little bit at the intersection of biology and engineering. So we definitely do take a team approach to science. And so we have people in the lab that are more from the biology or neuroscience side and others more from the engineering. And so what kind of an experimental day looks like, a lot of what we do is involving cells and cell culture. So you saw in the video one of the people in my lab going in and taking the cells out of the incubator. So growing cells is very much like growing house plants or something. Do you kind of check on them and how are they doing? Do they need more water, more nutrients, that type of thing? And then we make sure that the cells are really healthy and in a good place for our experiments. And then we have so many different types of experimental approaches that every day is a little different, right? And every day kind of brings new ways of looking at things. And I think that for us, we've really enjoyed looking at biology through the lens of using some of these engineering approaches because we've found out things we wouldn't have otherwise.
Very cool. Thank you so much, Dr. Flanagan. I now want to ask the same question to Dr. Mednick. What is a day to day in your lab? What does it look like?
Well, the video was great. Thank you so much for inviting me, and thanks for having our lab always be such a big part of the CNLM community. It's always really great for my students to have such a great community to associate with. So the video really did a good job of describing it. We have people, we study human subjects, and that has a whole bunch of work that goes along with just getting people to get into the lab to become participants in the experiments because you've got to go out and go get the subjects and bring them in, right? So there's lots of putting posters up all over the place. And then you've got to do a lot of screening of subjects. So phone calls and internet, questionnaires that they have to fill out. And then once you get a cohort that you think is a good one, then you have to do a whole consent form thing with them. So there's a lot of preamble to actually getting to the experiment. And then on the experimental day, it could be a nap study where people get tested on cognitive tasks in the morning, and then they get a nap with EEG, and then they stay in the lab after the nap, and then they get retested in the same cognitive tests, or they go to the cavern lab and they do their spatial navigation or whatever it is that we're doing it. Or we do an overnight study. And those are people get there in the evening and then they spend the whole night sleeping in the lab. And we have um, uh, testing there in the morning. And so that's like, the general protocol, but this can be done in older adults. It can be done. We have a big study we just finished looking at women across the menstrual cycle. And so people are in the study for months, and they come multiple times with it. One person, we track them across many, many times in their menstrual cycle. And so it's very involved, and so therefore it takes an army. We really have a very large group of we've got, a bunch of graduate students, and everyone has their own study, and then each graduate student has sort of, say 6 minimum undergraduates that work with them, and the undergraduates learn so much, right? They learn how to do EEG, they learn how to do cognitive testing, they learn how to deal with subjects like, experimental subjects, and even then how to process data. And then a lot of them go on to present at conferences. So there's a lot of mentoring, there's a lot of human interaction, and teaching going on in the lab while we're also doing the experiments.
Great. Thank you so much. So it seems like in your case, your experiments do require a ton of scientists. I just love that question. How about you, Dr. Krichmar? What is a day to day in your lab
Yes,
looking
the
like
loneliness
these
of the
days?
roboticist. No, we don't. You don't require a ton of roboticists. Depending on day to day, it can change. So if it's kind of an AI project, there's a lot of designing and programming going back and forth. With one or two students. Um, and then a lot of programming and running things in the background. When it's the robotics work, it takes a few of us, and you have to hope to phase the moon that the robots are behaving well. A lot of times, like the video nicely showed, sometimes we're indoors and it's a little more controlled. Um, Sometimes we go out into Aldrich Park and other places with our robots, especially lately, to kind of get out of the lab and into the real world. And then anything can happen. Uh, between different weather conditions, and then also, interacting with those robots that are delivering your coffee, and then students who are curious. So yeah, every day is different. The team is great. Sometimes, um, we need like helpers and stuff, and then so a group of us will help with depending on the experiment. Sometimes it's a lot of there's a lot of programming and engineering. I think the video nicely showed students on their laptops and bring up CAD models and designing materials and things. So yeah, so it's fun in that way. It's always refreshing and different, and every so many months we're starting a new project and we go in a whole different direction.
That's great. Thank you so much. Yeah, I think being a scientist, I've learned how much it can be both a team sport and an individual sport, and often it's both at the same time. You're managing your teams and you're working with all these different people, but then you have your project that you're kind of zoned in on, focused in on, and all the grad students have their own projects. So yeah, it's an interesting way of dividing up the work. But I want to go into a little bit more of the individual rationales and motivations and ask you each what got you interested in neuroscience, for one, and what got you interested in your specific research topic? We had a lot of questions from some of our younger audience members. How do I know if a career in neuroscience is right for me? And how did you get to where you are? So I want to invite you each to speak a little bit on that. And I think we can start with Dr. Flanagan. Once again, we can just keep the same order.
Okay, great. Yeah, no, that's an interesting question. And I know for me, it was kind of a gradual process. So I started out being really interested in psychology, and I also really liked biology. And as I went sort of further down the road of both of those, especially in college, I realized that I really liked the intersection of the two. And so that brought me into neuroscience. And then, going to grad school in neuroscience, you continue to get sort of further and further honed in, and you realize that in some ways, scientists end up studying scientific questions at different levels, right? So I think actually some of the people on the call here are looking at kind of whole body functioning, sleep, or how you think about cognition and adapt that into robotics. And then for myself, I really boiled down to cell biology. I really like thinking about things at the individual cell level. Other people go further still and just look at, DNA or molecular aspects. But I think it's important for each individual person to really figure out what makes them really curious. So for me, trying to understand how, for example, neural stem cells during development go from just, these single layer cells to creating the enormous complexity of your brain and spinal cord is just fascinating. And so every day when I think about that, I'm like, wow, how does that happen? And it really is motivating to me. So I think, you know, thinking about how these questions really spark your curiosity is important.
Yeah, absolutely. I totally agree. I am a fellow cell biologist, so I understand the passion behind like how are these interactions creating such absolutely complicated things. Yeah, it's so exciting all the time. Thank you, Dr. Flanagan. And now I want to ask you the same question, Dr. Mednick. How did you get where you are, and how did you decide this was your path?
Thanks for the question. I was it's my second career path. And so I used to have a much a my first childhood idea was that I was going to be an actor in theater. And that was it took up until I turned like 20 22 or 23 before I started realizing, like, well, maybe 24, 25, when I started realizing, like, even though it's an amazing life, it's sort of not um, stable enough and intellectual enough for me. And so I started just getting um, internships, and I was working in a mental hospital, and then I was working with people with schizophrenia, and I got really excited about the brain because when you look at people who are sort of, having mental illness issues, and you realize, like, well, there's something very different about what's going on with this person's brain than what's going on with my brain. And I got really excited by that question. And it just sort of, took me to the idea of I should probably study this and go to graduate school. And then um, and then how sleep happened was that I was looking for some kind of project to do, and I just happened to go to a lecture of this guy named Bob Stickgold, and he was just beginning his research on sleep and memory, and I just I felt like I had a kinship with him and his character, and I also thought how fun was the topic. And then we met and we really, got along well. And so it was sort of, it's that idea of like go to a lot of different talks, even the things that you don't think that you're interested in, because things will suddenly spark you in a way that you may not be aware of. And just so you can be open to things um, and put them together, and put ideas together that you didn't think were there before. So
Yeah, that's very cool. Just poke around and see what clicks and what resonates. But I really like that. And we're all trying to forge a brand new path. That's the crazy thing about being researchers is like we're trying to do something no one's ever done and ask questions that no one's ever asked before. And learning how to do that, it takes so much curiosity and really building that skill. So thank you for that. And now I want to ask you, Dr. Krichmar.
Yeah, like Sarah, I had a circuitous past, but I didn't have aspirations of being an actor. But yeah, I did my undergraduate at UMass, and one of the last courses I took there was artificial intelligence. I was too ignorant and naive at the time to know what the person was teaching was computational neuroscience and neural networks. I had a lot of student loans, so I had to work for a while. So I worked as an engineer for several years, and then I kind of wanted to go back and just learn more about AI. Um, and this was in the late 80s, so I went to do a master's, and at that time, what AI was something called expert systems, and it had nothing to do with the brain. And for me, it was just boring. And one of the last courses I took there was neurobiology in a psychology class, and that got me really excited. So then I continued taking courses. I had more loans to pay, so I was working as an engineer at IBM in Maryland, and then I just took go full forward in academics and got really into it. And so then when I was looking at what to do next, I wanted to combine what I did as an engineer, which was um, a lot of real-time and embedded systems programming, so making things move, reading from sensors. And I go, oh yeah, that's what a robot does. And lucky for me, a postdoc opportunity in San Diego happened. That was my dream job. They were just doing exactly that. They were using robots as a surrogate body to test large-scale brain models, and that just was fascinating. So I worked there 10 years as a dream job before um, deciding I wanted to set up a lab of my own and do the same thing, which happened at UC Irvine in 2008. Yeah.
Very cool. I cannot imagine being so early to the AI wave and seeing what's going on now. That must
It's
Did you see that coming?
crazy. No, I mean, AI goes in what's known as these cycles. And so when I was at my job at Raytheon, I said, I want to do AI. And they said, and IBM too. And I go, AI is out. No one's funding AI. So is, a winter for AI. And then the last several years, it's just been crazy. It's like nothing I've experienced. Yeah. So I've been around AI in one way, shape, or form for a long time now. And it's kind of exciting and scary to see what's going on right now.
Yeah, I can only imagine. Actually, we got a few questions about how AI is impacting everyone's work, and I want to circle back to that later with everyone, because I bet everyone has a different, sort of interpretation of that. But sticking to the scientific process a little bit before we get there, we had a couple questions that I thought would be good to ask specific members of our panel that were submitted by the audience. One is for Dr. Mednick, and I thought this was a pretty interesting question, which was, how do the neurophysiological methods you use in your lab to assess sleep compare to wearable devices that measure sleep quality, like a smartwatch or an Aura ring? Yeah, just maybe that can make it a little bit more concrete for us, these differences.
Yeah. So we are really interested in both, because we actually do some studies a lot of the studies we do in the lab is looking at the EEG, and the EEG is measuring the electrical activity in the brain on this massive scale, right? So how very large amounts of neurons are all firing together. And so you get these really big waveforms that you can read pretty easily in the EEG. And that gives us a lot of information about brain activity, and that's really exciting because we're neuroscientists, so we wanna be thinking about, brain mechanisms that are at the root of these behaviors that we're studying. But there's also a whole branch of our research that's interested in how real world people are out there, and how sleep is affecting their behavior, and the only real way you can do that is by using wearables. So wearables are a wonderful tool that can be used in science. Here comes my son. Um, and he, and so this is, yes, but you can just walk through. Sorry. He's continuously going in and out. Um,
We have a guest.
yeah, he should just answer the questions for me. Um, so so there's certain things that you can get from wearables, and it's definitely you can get really good measures of total sleep time. You can get measures of how long, sleep latency is pretty good. You can get how restless you were during sleep. Of course, what they're now trying to claim is that they're also doing sleep staging, and that's where things usually fall apart. There's better and better algorithms because not only are they using motion, but they can use your heart rate, and heart rate is a very good metric for sleep stage characterization. But they're still not um, they're still not very accurate, and they're a little black boxy about how their algorithms work. So that's another issue. Is sort of um, it's hard to necessarily know what they're doing with the data to come up with, "Oh, you've got, 20 minutes of slow wave sleep." Like, "Oh, interesting. Where'd that come from?" So, um, I think in a way, the real world experience of having a wearable that tells you, a sleep score or how much REM sleep or slow wave sleep you had is great. I think that that is what has brought sleep into the modern world. And why sleep is suddenly becoming interesting is because now people can actually measure it themselves, and I think that people feel really empowered by getting a sense of their own, um, how their sleep affects their functioning on a day-to-day basis. So those wearables I think are awesome, and you can get really large scale data from them as well. So if you want to do population studies, I think they're still not totally at the level of EEG, but I think that they're getting better.
Great, thank you so much. That also addresses a question that we had submitted about the accuracy of the wearables. So thank you. You get two birds with one stone there. I would like to move on to Dr. Krichmar again, and ask, what is the potential for robots impacting our lives? And that's a big question, broad, but I just want to hear your take on that.
It is a big question. Besides they're delivering coffee and Panda Express on our campus, uh, I think the biggest near-term impact will probably be self-driving vehicles. Like a lot of the, and I haven't done this yet, but a lot of the Waymo, the technology for self-driving vehicles has really come a long way, especially the robo taxis. There's a whole bunch of issues that need to be resolved, but still, um, there's a lot of hype around humanoid robots, which I think at this point is hype. I was out to dinner with a couple people who are experienced with startups, and they go, "You should just tell someone you're making a humanoid robot, and they'll be throwing billions at you." Um, so I think it's a long time before we see, unfortunately, which I think will be wonderful, robots taking care of the elderly, taking care of the impaired, helping around the house. It's got a long way to go. Um, the things that are more near term, manufacturing has really become roboticized. Um, and the other thing that I'm really interested in is exploration. So I think a lot of the Mars rovers have been really successful, and there's going to be more autonomy, more AI on these things, more biological inspiration for these things to explore, not just space, but deep sea, lots of areas that are hard to reach for humans or just too dangerous for humans. So there's a lot of avenues, but it's still kind of behind all the AI models that are taking over. There's still a lot to do, which it's a difficult problem. So for as a researcher, it's a great problem because there's a lot of interesting algorithm problems, material problems, design problems. So that makes it an interesting field to be in right now.
Very cool. Thank you. I think it's really interesting that you say that the more problems, the better as a researcher, because I think everywhere else, that's the worst thing. You just want it to be able to be implemented fast. But for us, we're like, yes, lots of stuff to figure out. Okay, now I have one specific question for Dr. Flanagan here. And it's what, if any, progress has been made in replacing damaged brain cells with new brain cells through stem cell usage?
Yeah, so thank you for that question. I think it's a really interesting one. And honestly, I think we're at a super interesting time in terms of thinking about stem cell therapeutics. So there's a lot of research that's had to go into this, right? So we had to figure out, what were the capability of these cells? And then we had to figure out what cells were the best ones to implement in various disease models or to treat various neurological conditions. So a lot of that work has been going on for, maybe decades now, but it's really cool because it's led to a lot of clinical trials that are now finding significant impacts of stem cell therapies in neurological conditions. So maybe I'll just talk about two, neither of which I'm involved in, but I think they're super exciting. So there's one that's for Parkinson's disease, and this is one that has been led by Claire Henchcliffe, who's here at UCI. And so the idea there is in Parkinson's disease, you're just missing this particular type of neuron that makes a particular neurotransmitter called dopamine. So the question was, okay, could we take these stem cells, turn them into, some that type of neuron that would make that type of neurotransmitter, implant it into the correct place in the brain? And they've just completed this really nicely designed phase 1 clinical trial. So clinical trials go through these different phases, right? And so phase 1 clinical trial is, okay, we're going to test this therapy and see if it's safe because, of course, the first thing is, do no harm. And then phase 2, you move into starting to test efficacy and how much benefit could you get from a particular therapeutic. So in this phase 1 clinical trial, first of all, the therapy, the stem cell derived neuron progenitors was completely safe. There were no adverse events. So that was really a relief. Very small trial, only 12 participants, but that's how it was planned. And even within that, they could do imaging to tell that the cells were surviving at least a year and still able to make dopamine, which is fantastic. And their initial tests of whether the Parkinson's patients, got any functional recovery were really promising. So they did see some functional recovery. So now they're moving into the next stage of those clinical trials, right? So they're going to have many more than 12 participants, and they'll really start to see, okay, so how much benefit can we get from these transplanted cells? So that's one. And then I'll just tell you a very short story about another one on Huntington's disease. So that's work that's really been spearheaded by Leslie Thompson here at UCI. And same sort of thing. So you've got neurons that are lost in a neurodegenerative condition. And so they've been finding in their animal models of Huntington's disease, when they transplant in the stem cells, they can see them differentiate into a lot of different types of neurons. Those neurons connect with each other and with the host brain tissue, and they get functional improvement. So they just got a big grant to start clinical trials for that. So, we're all sorts of different groups are kind of at different stages of the pipeline. And so the research in my lab is trying to understand how the neural stem cells decide which of these cells they're going to differentiate into, and how might we use our understanding of how that occurs during development to help guide their differentiation into particular cell types in a transplantation regime. So we've got everything going on from, these very sort of basic studies to just starting the clinical trials, and then clinical trials that have already shown to be safe and are now moving forward to determine efficacy. So I think it's a super interesting time in terms of stem cells for clinical applications.
Thank you so much. That's really exciting. Thank you for bringing this to really direct applications to our attention. It sounds like everyone has a lot of excitement and energy in their fields right now. Like a lot of things are, right on the precipice of really, I don't know, taking on much broader relevance. So that's incredible. Thank you so much for sharing that. And I do have one more question for everyone before we move on to our next section, which is about failure. How have you had failed experiments? How do you move past failure as a scientist to keep learning? And how do you make sense of inconclusive findings in your research? Let's start with Dr. Mednick again here.
I love this question. Um, yeah, it's such a good question, and we actually have talked about this in lab meeting, as a lab because I have this sense that science is like 90% failure, and you have to really get a tough skin about it and have a, curiosity mindset around the whole process of science, because it's it's very competitive, number 1. So you could spend, just at the level of, getting jobs, getting grants, you spend months, years doing, creating projects and writing grants and going for jobs, and, you'll not get like 90% of those grants, right? And maybe 80% in good years. But right now it's like 95% of the grants don't get funded. So it's a very tough time. And so there's like a really intense amount of uh you know I think I'm trying to figure out like what is my value if my value of of my science is being valued by this, you know, in this way that looks like I have no value, right? Because like I'm not getting grants or I'm not getting a job or I'm not getting all this stuff. And because we have this kind of external need for for peer review or funding from external sources um getting giving people, people have to give us jobs, we can't just go out and be an artist and like live in our studio. And I mean, obviously that's also really hard, but we need a lot of feedback, and it's very sometimes can be very hard to get the feedback that you're looking for or to have success. And so my and experiments, I don't have such a issue with experiments being failed because I don't think that there's such a thing, like, because if you do a good experiment, no matter what, there's gonna be an answer. It's just the idea, um, of the answer is that it's probably not what you thought it was going to be. So it's totally fine to have a really strong hypothesis, set up a study to to answer this question, but set it up in such a way that no matter what the answer is, it's going to inform you. And then you kind of give up on what you thought and just let it tell you what it is. So to me, that part of science is less of a tr- worry. It's more the day-to-day in and out of, like, getting a job, getting a grant, getting a paper submitted, published, all that kind of stuff. That's, I think, where a lot of the failure comes in, and how you to me, like, the way I've really worked with that is just to try to keep an eye on, like, how do I value the work? How do I value myself? How do I value my students' work? And not, and try to keep that really clear because there could be many months, days, weeks, years where the external world doesn't necessarily give you what you think is the same level of success that you deserve or think you want.
Yeah, absolutely. So we have two kind of failure to contend with, I guess, here. We have the experimental failure, which you say is not really a failure. You always are going to learn something, but then also the difficulty of securing funding and how thick of a skin you have to develop because you're going to apply for these things over and over and over. And actually, we had an audience submitted question that I think might be a good follow-up, just to ping right back to you, Dr. Mednick. Would you say luck is required in your job?
I don't know about luck. I don't know what that is. I don't know if luck. I don't know about that. No.
Yes.
Hard
My
enough for me to
experience
say
is a
yes
lot of,
also.
yeah, there's a lot of luck, a lot of randomness, and you just keep trying. Sooner or later, you break through. Yeah.
Well,
Uh,
is that luck or is that just you just persevering through a lot of failure? I don't know if that's luck. I think it's just working hard and
Well,
I
I
think
Persistence.
think luck favors the prepared, right?
Yeah, that's a good quote. Yeah, and yeah, there's luck of the draw. There's, a lot of people eyes looking at it with different backgrounds, different moods for the day. So, there's a lot of stuff like that. But both of you are right. You kind of take what you can that's constructive and try again. Yeah.
I guess true. Like with there's 3 grant reviewers, and you're lucky if they've slept well and had enough food and sleep and coffee to be like coming at the grant in positive way, and that you're the first grant they read and not the 8th grant they read. Yes, that there is luck in that. I think that that's true.
But maybe in the sum, you're not entirely relying on getting lucky. I want to now ask the same question about inconclusive findings, failure to our other panelists. Let's ask Dr. Flanagan what your take on this is.
Yeah, I actually agree with a lot of what Sarah said in the terms of a couple things. If you think about how you're designing an experiment, you can really map it out where, yeah, you're going to learn something either way, right? So either this is going to happen or that's going to happen. And if this happens, we think it might be because of this, and if that happens, we think it might be because of that, right? And either way, you've got a jumping off point for your next experiment, right? But even with the best plans and thinking it out and all of that, sometimes just technically things go wrong. There are certain things. The one thing that we end up dealing with a lot in terms of ourselves is sometimes things just go awry, and we don't know why, right? Like we've gone through our list of, oh, it could be these things, and we've checked them all, and you have to decide, well, okay, how far am I going to go down that rabbit hole, right? Or do we say this is not really the main thing, and we'll just get a whole new batch of reagents and just start over, which is often what we end up doing. But so in that way, I think that, not getting too caught up in the failures or not getting too distressed by them, because they're going to happen, and we just, find our way out of it and move forward. Other than that, the only thing I think I would add is that sometimes I feel like being a scientist is a bit like being a detective on a case, right? Or Sherlock Holmes or whatever. And you're going down a particular path, and you think you've got your suspect, and then you see something, and you're like, oh, they have an alibi. That's not right. All right, let me think about other possibilities, and you go down. So if you have that mindset where you like that, and you like coming up with new ideas when what you initially thought didn't pan out, then science is for you, because that's really a lot of what we're doing day to day.
Amazing. I really appreciate that take. Yeah, sometimes I've done cell culture before, and there's every scientist I know is really into some voodoo. Like there's especially mass spec machines. They always have a little saying that you have to say before you use the machine. Like things are finicky. But yeah, so there's all sorts of, you got to really keep going, right? Dr. Krichmar, do you have a take on this question?
Yeah. Well, Lisa and Sarah have done a good job of answering, kind of the broader, the way we work, it's a little different. So there's not as much well-planned experiments. A lot of it is trial and error. You have an idea, you want to run with it, kind of what the model is going to be like, or what the robot should be doing. But then things never work out, and you keep going back and forth. And it can be frustrating for students who haven't gone through that cycle. And then at some point, things just come together. And I like the robotics work for a bunch of reasons, but when an algorithm or an idea works on a robot that's, you know, in the real world with all the noise and stuff, uh, and it works, then it's really robust. And it seems like once you hit that point with us, then you can throw a lot of things at it, and it still stays stable and works. The other thing, going back to luck, there's at least with the robotics work, there's this serendipity. I always tell my students to watch the robots while it's behaving, not just turn it on and leave the room, because you kind of see these aha moments that all of a sudden, like, oh, I didn't expect that. Why did it do that? And so something emerges, and that's where it gets really exciting. And I haven't done an experiment or seen one of my students' experiments where there wasn't something that kind of emerged we weren't expecting that was the coolest thing, the thing that I thought was the biggest finding of that.
Yeah, paying close attention, you can pull so much more out of these experiments. I've looked at images before, and I'm just trying to quantify one thing in my image, but then I noticed like something's a little weird with these cells, like they're kind of shaped a little bit differently, and then there's all these different rabbit holes you could go down. So maybe you get lucky in that you're asking the right questions, but maybe you also are just doing a good job observing all the things that you could observe. And yeah, whether that's luck or not, I think is a really
Well, Lisa said
interesting
that
thing to contend with.
luck favors
Yeah.
the prepared, so that's actually a good way to think
That
of it.
brings
Yeah.
us to AI.
Uh-oh.
Yeah, do you guys wanna get to I think we can actually get to that question now if you guys are interested in talking about it, because I know a lot of people are wanting to hear about how AI is affecting everyone's research.
How should we start? You want to do
Okay, let's start with Dr. Mendick since you brought it up.
Yeah, I mean, I see a big shift in the scientific educational process with AI, right? I mean, that's not a surprise statement. Um, but exactly, what Jeff is saying is you need to sit there and watch things happen and be an observer in a way that you're you're looking for novel things that you don't know about and that it takes a lot of time and that um gosh, it just it is a very work heavy sitting there doing it yourself hours by yourself focused on something that may or may not happen process. And in my lab we do a lot of that. We just stare at EEG, from sleep scans, constantly. And there's a lot of AI now that will do all your sleep scoring for you, right? That will analyze data for you and that will, actually write your papers for you and will also summarize all papers that are in your field for you and outline things and let you sort of you could just send it some ideas and it can write a paper or write a grant. So and to me, um the discovery part of science is exactly what Jeff was saying is so many of the studies that we do, um, the discovery comes the interesting thing is not the thing we set out to look for. It was the thing that came up because we were prepared, right? We set up the study well, and we thought it through, and we made it interesting study. So nature had a way of coming through and showing us it herself, something really interesting. But if you're not there to see it, and you're not really paying attention to all those you know, very tiny details and things that are novel that you don't expect, and that an AI system is going to be just looking for regularities or looking for things that you already told it to look for. You won't see those things, and so we're a little analog in my lab because of that. I try to get people to write their own papers, and I try to get people to score their own sleep and analyze their own data data point by data point so that they really can see things. And I am concerned about that process is going to be slowly taken away or quickly taken away from students.
So a bit of more of a cautionary tale there. Yeah, I think that's really interesting. How about you, Dr. Flanagan? How are you seeing AI maybe impacting your work? Are you integrating it into your lab's workflow at all? Where are you at with it?
Yeah, I think, to me, I sort of feel like I'm grouping in my mind, AI things into sort of two camps, right? And one camp is there are things that we as humans can do, right? And you can use AI to kind of do that for you. So that's a little bit, I think, like what Sarah was talking about. Like my students could sit down, in front of a blank page and start writing up their paper, or they could ask AI to do it, right? And that part, I'm more keen on the students doing it, right? It's partly like Sarah was saying, so that they are actually really thinking it through, because then they might come up with new unexpected insights. But also, writing's hard, and if we as humans don't practice it, we're gonna get really bad at it, and I think it's an important skill for people to have. So, I'm less keen about AI in that type of context. However, the other part of AI that I think is actually super interesting, and I think is something we can start applying more and more, is dealing with really large data sets that we have a hard time sort of parsing through as individual humans, right? And the reason I say that is we've had, how many years? 75 years of really excellent scientific research that has delved into all sorts of different, cellular processes of the brain, things like that. And um, coupled with all the data that's coming from electronic medical records and things like that for patients that have different diseases and different conditions. And so finding ways to kind of mine that, pull all that data together and look for, associations and pathways and things that might not be so easy for us to find as we're just looking in our one little area. That I think is actually really exciting, and an area where AI can kind of push research forward by saying, hey, have you noticed that there's this similar association of this pathway in both, stroke and, ALS? And no, in fact, we hadn't noticed that. And then we can do experiments to really test whether that's something that could be addressed therapeutically. So that style of AI, I'm really excited about.
Very cool. Thank you. Yeah, more using it as a tool and something that can help us rather than having it be something that does our work for us. Yeah, it's I'm a grad student, so I know how hard it is to learn how to write a paper, to learn how to read papers efficiently, but I'm so glad I've taken the time to learn how to do that, because if I compare myself, at the beginning of my PhD versus now, it's like I learned a whole new language. But if I had just had I haven't been here long enough that I didn't have access to AI tools when I started my PhD, so there was no choice. But if I had been using those the whole time, then, I don't think I would be able to read the papers like I am now. So, but I have also used it to help me code, because I didn't know how to code when I started my PhD, and it helped me learn how to do that. So yeah, pros and cons everywhere. Dr. Krichmar, now I would like to hear your take on the AI question.
Sure. And interesting, you mentioned coding. I mean, that's become quite good. I mean, it makes sense that it would be quite good at that. And as long as you know the fundamentals, it's actually quite about the how to do coding, so you can understand the code it spits out. But I've been pretty impressed with that. Yeah, I'll talk about it as used in our lab. There's a whole other, if you want to get into it, societal issues. I have thoughts about that, of how AI is affecting society. But in our lab, it used to be, like I said to all the students, no AI. We're not doing that. We're studying the brain. We got to make brain models. Um, but I got a lot of talented students that were interested in AI, and they wanted to promote their careers, and that's what they wanted to do. So we went back and forth, and the tools were getting so good that I go, all right, I can't ignore this. So the last several years, we've been doing a lot of the state of the art AI models. I should say my students are. I mean, they're really talented in this. And what I've kind of come to an agreement with them, and I think it's quite powerful, and it's becoming a field called neuro AI. So you can take these AI models and you can either cast it in a problem that people or animals have to solve, and then look inside and say, do I see things in the model that resemble brain recordings? And you can also put some biological inspiration, like different anatomy or different kind of modulation, and different kind of learning rules that are more bio-inspired, and see how that actually changes or improves AI. And so I think there's been a lot of traction in both of those for the field and my lab. So we're doing a lot of that nowadays. Um, but I think this is a little bit of societal stuff. There's talk about artificial general intelligence like humans. There's talk about machines that become conscious or self-aware. I don't see that happening anytime soon or ever. It's not the same as biology. Biology is doing something very different. And the robots are getting a little closer, but they won't, because if you do the robots right, they're kind of interacting with the world, but they aren't feeling or living the way biology is. So I don't see that match, and I don't see that as an issue, except for people that are convinced that what the AI is telling them is something sentient. I don't think AI is, but it can be convincing. So that's a concern. I guess I was getting to societal stuff. Sorry. But yeah, AI is a big part of the lab, and now I'm embracing it as long as it fits within, our mission of neuroscience and cognitive science and solving those kind of problems with it.
Great. I really appreciate the array of different thoughts that we had on this because I think you guys all touched on different important aspects of AI in the modern research space. So thank you all for that. So now I want to move into a few questions about the importance of your work, the importance of scientific research, the importance of scientific funding, which has been quite a hot topic in the media and for us especially. And I want to start this off with how do you decide which questions to ask? We talked a little bit about this with the failure question, but how do you decide what to ask and which ones to pursue? Because there's all these rabbit holes we can go down. How do we make decisions about what to actually pursue? Do you want to start again, Dr. Mendick?
Sure. So there's things that you pursue because there's funding for that thing. And then there's things that you pursue. I'll use I words. There's things that I pursue and that my lab pursues because uh we're just curious, and that you see this kind of building up of ideas around something, and it almost just feels like, that the experiment just comes in and sits and just plops into your lap as this obvious thing that you just can't ignore, that's the next step. So, um and those that the idea of pursuing things because there's money for it, and pursuing things because it's a curious idea that seems like something, how could you not do this? Those are two different paths, but they often overlap because the thing that you're curious about, there's a zeitgeist around that thing, right? So there are people who are also curious about it, and therefore there's usually going to be some sort of funding that will support that thing. But then other times, you know, you I have gone towards um, like particularly like in the you get money from the Department of Defense, and you sort of have to couch those kind of things that you're curious about into questions that you're really not curious about. um and so but it works because everybody kind of gets what they need. There's the military gets the questions that answer that they want, but then you get to also answer the questions that you want. So to me, it's a lot about, and also just as a mentor, I am usually trying to find a topic for people, that is something that is coming out of the lab that we're already doing, that's already a general area, but that I can also see like, oh, this would be so perfect for this new grad student because she's really interested in this question, naturally. So I try to also make those connections. But that's sort of the general way that these ideas get pursued.
Great. Thank you so much. I would like to now direct the same question to Dr. Flanagan. Is it different? Is it the same?
Yeah, I think that for a lot of the work we do, it's a little bit of a combination of sort of, where is the field going, or where are the gaps that you identify? And then where do you feel like you can make the biggest contribution, because there's a lot of people working on these different areas. And so what is your kind of niche? What is your approach that's really going to bring something new to the table, or figure out something different? So that's part of why my lab has ended up at that interface of biology and engineering, because we've been able to use these engineering tools to discover new things about neural stem cell biology, and that's led us to understanding more and more about stem cell differentiation. The other thing that for me has been really important in terms of thinking about what scientific avenues to pursue is I really like being in the Department of Neurology. I get to go to grand rounds and hear from my colleagues, where are we currently in the state of treating different neurological conditions and diseases. And a lot of times for me, that gives me kind of ideas like, oh, we're actually doing this thing in the lab that could be relevant for this, and we could move our direction in this, our research in this direction, and maybe ask a question that could be relevant for developing something that could move into clinical practice. For me, I find that really motivating, and that helps to direct how we pick what research questions.
Thank you. And just for a little bit more context, at UCI, we have the Department of Neurobiology and Behavior, which is what I'm in, but then we also have the Department of Neurology, which lives in the medical school. So there is a lot of distinction that maybe, people aren't aware of. And the medical school has more interaction with clinicians and doctors. So I think that's really cool to integrate that into your work and deciding how you answer questions. So very cool. How about you, Dr. Krichmar? How do you decide what to pursue?
Yeah, it's a great question. I've always tried not to follow everyone else. I get bored when I'm in the field with everyone doing the same thing. And you see a lot of that, especially when something gets hot, then the funding agencies and everyone wants to go that direction. So, we don't have enough resources to compete, especially if you compare with AI nowadays, with OpenAI or Microsoft or Google. Um, I always tell my students, especially if they want a career in academics, take a look and see where there's kind of room to actually go in a different direction. It's not completely out there, um, but it's not where it's crowded, everyone else is doing it. And then maybe you can, be the person that's doing this work. And that's sort of how I pursued it. That's why I got into all this neurorobotics. Um, the other thing when it comes to funding or just reasons to do what we do, there's two. I mean, one's the basic research using the robot or the model as a tool to understand how the brain works. The other is more practical and applied, using what we know about the brain to inspire applications that might be useful one day. Since we're basic research, except for a couple times, we haven't attempted to start a company or make a product, but hopefully someone reads our work or gets inspired by our work, and that leads to that. Um, yeah, and so that's the basic idea. The other thing is, I've been around long enough. I don't necessarily have to fund a large lab. So a couple of the robots you saw in the lab are just a whole nother direction that we said, hey, let's look at this. Instead of looking at mammalian brains, what about other brains? Because of organisms that are really intelligent. And that led us to cephalopods. So one of the robots was based on an octopus, and another robot was based on this related cephalopod called a cuttlefish. And so that's been really exciting, just to kind of go in a completely different direction, and kind of explore like that. I don't know if it'll come to anything, you know, um, as far as, a big money maker, but I think there's already interest, and it's an open area where there aren't that many people looking at it.
You bring up moneymaker, and that perfectly segues into my next question. And a couple of you brought up funding, and that is definitely a hot topic. So I want to ask, and I think you guys all need to pitch me a little bit because I want this to make sure it lands with our audience. Why is your work important to fund publicly, with public funds, with taxpayer dollars? What is important about it to the everyday person? And you could talk about your work specifically, or just a little bit about funding of scientific research in general, however you want to approach the question. And let's start with Dr. Mednick.
Um, Yeah, it's a good question. So, why should we be putting public funding or private foundation money into science? So there's this really big macro level question, which is that I think humans have a deep need to understand why we're here and, who we are as humans. And so, um, I, as much as you can, there's many ways to answer that question. I do think that science is a really great one. It's development of knowledge around what makes humans develop into adults and what makes animals develop into their own adult form and what is the origin of all of these different things from the lowest level to the largest level. I think that science is it's one way to answer that, and I think it's a um valuable it's a very valuable way to answer it because it's endless, and that's another part of it, that there's an endless amount of questions because we can only answer, as individuals we can only answer a certain amount, right? And something that Jeff said made me think, like, you never really know what value one study is going to have, and you never really know what value you as a scientist are going to have. You don't know what a discovery you're going to have when you're in your 20s in graduate school may affect somebody 100 years later, right? And there's a lot of that goes on in science. And so I think, for that reason, science should be funded, and it's a super valuable thing to do with your life. And then what we in just to quickly talk about what we do in my lab is, we are studying something that is a basic need. Sleep is a basic need, and it's a very under researched area. It's kind of only really started to gain traction in science very recently. And so it's even though it's something that we do for 20% of our lives. So it's something that really deserves a lot more attention. And what that also means is funding. And in particular, in medical research, doctors, when you're going into medical school, there's no course on sleep. People don't learn about sleep independently. It is really an under utilized resource in the medical field, and it's also underappreciated in terms of its impacts on psychology and biology and neuroscience and behavior. So I think sleep is a really big open area that could, have connections to a lot of different exciting discoveries in the future.
That's great. Thank you so much, Dr. Mednick. I I agree with literally everything you said. I would like to now ask Dr. Flanagan the same question, and you can take it broadly or talk about your lab in particular.
Yeah. Yeah, I think it's super important to continue doing research, and especially I'm going to talk about it more from sort of just the biology standpoint, right? Where the way that we end up with things that are in the clinic that are treating diseases is because we have basic research that is happening in labs across the country. And then that basic research, some of it looks really promising and it moves forward into some of those early stage clinical trials, like I talked about. And then we end up with something that can treat, millions of people, right? And it could help your aunt who had a stroke or your grandfather who's got Alzheimer's disease. And if we don't maintain that pipeline and keep making new discoveries, then we're just going to sort of stop where we are. We're not going to be able to identify new treatments for all sorts of different conditions that people are dealing with every day. And so, just to kind of give an example of an idea of how this pipeline can be, we have research in my lab. So we've been looking at this question of neural stem cells and how they form neurons and astrocytes and oligodendrocytes, the mature cells of the brain. But we've been able to apply those same techniques and technologies to studying glioblastoma. So, glioblastoma is an unfortunately really deadly primary brain tumor. And so, right now, the current standard of care is you have surgery to take out the tumor, you have treatment with radiation and chemotherapy to try to kill off any remaining tumor cells. But what happens, unfortunately, is that the tumor grows back and then it's resistant to further chemotherapy. So we're trying to understand those cells that are resistant to chemotherapy, and it turns out they're acting like stem cells. So they're expressing a lot of the same markers. And so some of the approaches that we've developed for stem cells, we're now applying to understanding these cells that are resistant to chemotherapy in glioblastoma. And so we found ways to isolate those cells. Just been doing rounds of sequencing, and we're finding common pathways between those and stem cell populations that are identifying new targets and new ways of modifying the behavior of those cells in patient tumors so that they can be now killed off by the chemotherapeutic. So, keeping that pipeline open so that we can continue these types of really, important discoveries that can eventually lead us to improving human health is, I think, critically important.
Great. Thank you so much, Dr. Flanagan. And yeah, something you touched on, I think, is how much things build on each other and like seemingly inconsequential findings can lead to something, so critical down the line. And every stage of research is so important to fund for that reason. Dr. Krichmar, I would love to hear your take on all of this as well.
Yeah, I mean, like Lisa and Sarah said, we're all doing basic research. It takes a long time. You don't know uh, where this might lead over the years, but the biggest discoveries and most important discoveries have come from basic research that was not as directed. I don't want to say 100%, but many times, and there's lots of examples in every single field. Um, in the public funding actually helps, not only buy equipment and resources, but also it supports our trainees, our students, and our postdocs. And I think the best thing that's ever come out of my lab is the students and postdocs that have gone on to do things, whether it was academics or whether it was in industry. They're going on to do great things. And I think the training they got through public funding actually made a huge difference in what they could do and the way they approach things. So I think that's not always, comes out. More specific to our field, the AI and robotics community, there is now actually, which is a good thing, a lot of industry interest and things happening there. But most of the discoveries and things that led to that came out of, our communities. You know, uh, People writing papers, people going to conference, people coming up with ideas, that all led to all the boom that you're seeing now. And it took years. I mean, like I said at the beginning, I started in the late 80s and AI was in its, I think, second winter. I forget which which peak we're at now, 4 or 5. So it took a long time for this actually to really become something that everyone's talking about and using. And I think that's would be the same for all fields. And then, as Sarah and Lisa probably know very well, when you write grants, you don't just write like your nerdy scientific stuff. There's, especially with National Science Foundation, there's a section called broader impact. And with NIH, which I'm not as familiar, but I've written some grants there is you have to put that in where you say, how is this going to benefit society? How is it, why would anyone care? And if you don't do a good job of that, you're not going to get funding. So you do have to convince uh, the funding agencies that this is not just navel searching or navel gazing. It's actually something that's worth funding by the public. But the public needs to understand it. It takes time, and it's not always the direction that you think it'll go. That leads to some great things.
Yeah, absolutely. Iterative process, slow process, but that's how we get from point A to point B in science. And we're winding down on time a little bit, but I just wanted to bring one more question to everyone. And maybe if we could have just someone jump in and answer this question, because I don't know that we have time for everyone to answer it. But I wanted to talk about trust in science. And there's so much information out there, so much data out there, so many people speaking as experts on so many different topics, right, on social media and and everywhere. How do you decide, you as a scientist, but also for our audience, when they're just trying to decide what they should trust, how do you know what to believe? And I know that's a huge question, but if anyone wants to take a stab at it to close this out, I would love to hear your thoughts.
Well, it's getting very hard. I guess that was why there was silence. I'll give it a try. Because I was thinking about this, some of the, doing a lot of reading your own research, but no one has time. Some of the sources I look for some things like the New York Times and Wall Street Journal, I'm sure there's other venues, have really good science and technology sections. And they usually provide, you know, names or references that I can look and go, hey, what's the details? Because journalists, want to make it so that people will read it, but they're very well done. So I trust that when I'm reading. It's going to be a big issue. There's a lot of hype out there. Um, there's a lot of noise, and social media can be like a rabbit hole, as people say. So I think just being well informed and picking sources that actually vet who they're interviewing and back it up with good science.
Yeah, I'm gonna add a little bit to what Jeff said, and I'll just add in the BBC. I feel like they do a really good job of describing science on their site. I always feel like it's easier to answer this question as a scientist than as somebody in the general public, right? Because as a scientist, you go and you look for the primary data. You look for, okay, somebody did this study and they compared, A to B. Let me go look at the facts, right? And look at how did they design the experiment? What did the data show? How often did they repeat it? And did anybody else in a different place do a similar study and find the same thing? And if all of that looks solid and looks good, then you're like, okay, I know I can believe that finding, right? But that's our job, right? And that's part of what we do. And we have we know where to go to find those papers and things like that. So I think it's easier for us to feel confident about the scientific findings upon which we are basing our own studies. But I think the answer is a little bit tougher, like you said, with the current landscape and how much disinformation can be out there. It's a little bit harder.
I do want to say we should do a better job as a community communicating science and do more of it. I think our voices need to be heard, and I think that's something that all of us probably should be doing, and myself included. Yeah.
Well, thank you all for being a part of this today, because I think that's how we start the dialogue. And we have to recognize where we're falling short. And if we're not communicating enough with the public, the first step is recognizing that. So I appreciate you all for being here. I wanted to leave us off with just one more little tidbit, which was, if there is a good resource to learn more about your research, feel free to tell us about it now or post it in the chat. And otherwise, I just want to thank everyone who participated today, everyone who was here, for being here, and for participating in our second How It's Made Neuroscience. We hope it was interesting and enriching for everyone who attended, and so grateful to our CNLM team and our panelists for helping put this together.
My pleasure. Thank you, CNLM.
Yes, thank you, CNLN. This has been really fun. Thank you so much.
Yeah, nice talking to everybody. Thank you. I'm putting our lab websites in here.
Awesome. Thank you.
Thank you, everyone. Have a wonderful night.